Good evening, everyone. Welcome. I'm Anna de Koninck. I'm the Executive Director of the Koch Institute. Welcome to our first Solutions Within Sight of the academic year. This year we are celebrating the impact of our signature programs. Next month we will highlight some more success stories from our Koch Institute Frontier Research Fund program. Tonight it's all about the Bridge Project. As you well know, Harvard and MIT are both well-known universities, typically assumed to be somewhat competitive, but generally considered among the very top universities and academic research institutes in the world. And on one side of the river is the Koch Institute. We are one of 7 basic National Cancer Institute, or NCI, designated cancer research centers, with its unique approach of blending science and engineering to speed up the rate of progress in our fight against cancer. And then on the other side of the river is the Dana-Farber Harvard Cancer Center, a large, also NCI-designated comprehensive cancer research center with 5 of the nation's top cancer hospitals. And until quite recently, despite being separated by a mere few miles, too soon, by a mere few miles and some muddy water, there were very few collaborations between these 2 NCI-designated cancer centers. So, and then, this is the Bridge Project story, a genius who is here tonight, called Art Gelb, first imagined the possibilities of a partnership between MIT and the Dana-Farber Harvard Cancer Center. Since its inception, the Bridge Project has been led by Tyler Jacks of the Koch Institute and David Livingston, Deputy Director of the Dana-Farber Cancer Institute. And they together have brought previously disparate institutions together to literally bridge the Charles River and bridge the gaps in cancer treatment. We are privileged to have some of these remarkable people here tonight. In our audience is one of our co-founders and Bridge Project leaders, David Livingston, or at least I was told he would be here. I'm not sure he's here yet. I do know the Red Sox are playing, so who knows? He and Tyler are inspirational and creative chiefs for this Bridge Project effort. Also in our audience tonight is Art Gelb. Without Art, there would be no Bridge Project. Art is an MIT and Harvard grad with a physics and engineering background. He is a member of the National Academy of Engineering and an entrepreneur, has been awarded numerous prizes and honors, and sits on many boards. He is a life member emeritus of the MIT Corporation, and he is a distinguished trustee of Dana-Farber Cancer Institute. It's my honor to invite Art to say a few words this evening.
Thank you so much for those kind words, some of which I didn't recognize. What I'd like to do is tell you a little more about my background to the extent that it's relevant to what I want to say. Then I'd like to share my light bulb moment, not the only light bulb moment in the Bridge Project, but this was the first one, I think. I'd like to talk about how I made some connections to try to make this thing happen. Talk a little about the Bridge Project. I'd like to also interject something about a trustee science committee meeting at MIT, which Harvard people will please not listen to. And then I'd like to comment about the economy award meeting last week. Background. So I did a multi-departmental doctorate at MIT many, many years ago, and I learned that in addition to the deep knowledge you get in any one department, really great things happen at the intersections, and you all know that. About 22 years ago, I was invited to become a member of the MIT Corporation, the governing body responsible for the Institute, and I've been involved with it ever since. So that's the MIT side. I kind of knew MIT as a student and involved with its governance. In 1999, I got to know cancer well and became a patient at Dana-Farber. A wonderful place. And as I was spending some time there, interacting, finding out what it was about. I made the mistake of raising my hand at a meeting and was invited to be chair of the genitourinary visiting committee, genitourinary oncology. So I did that for a while, and later on was invited to become a member of the executive committee of the board at Dana-Farber. So I got to know it pretty well. So that's background. In about 2006, I'm guessing now, I don't remember for sure, I had just left a meeting at Dana-Farber on the 15th floor of the Dana building. I was at the elevator waiting for it, thinking, what a good meeting that was. And I thought, where were the MIT people? Had they been there, this good meeting would have been a much better meeting. MIT brings a very different point of view to a great deal of what it does, beginning from technology, from engineering. It just has a different view of the world. I thought, I can't be the only person in Boston that knows both MIT and Dana-Farber well. That didn't make sense, although lots of people knew one or the other. But I decided then to commit myself to bring these institutions together to do joint research on cancer, and that was my light bulb moment. I was going to commit myself to do it. Didn't have the foggiest idea how to go about it. So the next business was making connections. So I went and saw Ed Benz, who was the president of Dana-Farber, and I said, Ed, we need MIT. And I gave all the reasons. And he said, you're absolutely right. But his body language said, you know, I'm running a large world-class organization. I'm kind of busy, which was perfectly fine. I then went to see Susan Hockfield, who was president of MIT, and I said, Susan, we need Dana-Farber, and gave all the reasons. She said, you're absolutely right. But her language, body language was, I'm busy, I'm doing so many other things, and she was right. So I set about to speak with whoever I could find at MIT, Koch Institute, at Dana-Farber, anywhere, to see if I could get people interested in making this thing happen. And I spent a couple of years walking the halls, talking it up as best I could, until one day, and I think this was in 2009, I walked into a room in which there was Tyler Jacks and David Livingston, both of whom were reaching out to each other. And I thought, bingo, there are the pieces. And that was the beginning of serious activity. The three of us talked about what could be done to help produce a joint activity. It turned out my wife and I decided to fund this activity, and in the first few tranches did just that to give it a head start. One thought that I had early was that any funds that come to the Bridge Project would be split 50-50. If MIT needed 60%, they would get 50. If they needed 40, they would get 50. And the reason is that I didn't want to see any intra-Bridge Project competition for funds. I wanted us to succeed as a group or fail as a group. And that's the way it's been ever since. You know, there aren't many places on Earth that have the density of supreme talent that exists within a few miles of this place. Across both sides of the Charles River, putting them together makes this absolutely unique in the world as far as I know. There are plenty of smart people everywhere, but the density of that intelligence and the drive to make things happen is supreme here. So the Bridge Project started, and it turns out that Tyler Jacks and David Livingston have been the Bridge Project. They've organized it. They've decided how to bring together terrific talent to work together, egos left aside, egos at the door, and make it happen. And it's been happening. Some great things are happening. One of the reasons I wanted MIT in the act is that it occurred to me that MIT would see things no one else would see because of the way it views the world. And you're going to see a project tonight that is exactly in the image of that, when you see a device that was envisioned here playing an enormous role, and potentially even bigger role in cancer research. That's coming later. I'd like to tell you about a talk I was asked to give at the Trustee Science Committee at Dana-Farber. I enjoy having fun, and there are certain things I can get away with saying many people can't. So I was asked to give this talk, and I show up at the meeting in a room full of people, no MIT people. It's a Harvard-centric community, and that was great. It's all the hospitals and so on. And I started out by saying, you're probably all wondering why MIT is in the Bridge Project. And I saw a couple of people nodding yes. And I said, well, I'm going to explain why. And when I'm finished, you may wonder why Harvard's in the project. There were a few disapproving looks. And I then went on to explain. I said, you know, biology has become an information science in the recent decades, an information science, and everybody agreed. I said, do you know where information science was invented? It was invented at MIT. The professor who invented was a true genius named Claude Shannon. When he did his master's thesis, it was considered to be the single most important master's thesis done in the century anywhere on Earth. If you read it, and you're in that area, you would agree. This man was beyond amazing, and he came up with the field, and the field is pervasive in everything we do. Computers are driven, communications are driven by the thoughts he had. So that's part of why MIT ought to be in the act. When I was finished with my presentation, David Nathan stood up. David had been a president of Dana-Farber, and he said, Arthur is exactly right. He said, you know, some years back I spoke with my colleagues at Harvard talking about sequencing the genome, and they all said it couldn't be done. However, people at the Whitehead Institute at MIT didn't know that, so they did it. David said, we need MIT, and that's how he ended his comment. And I'll conclude this comment by saying, and MIT needs the Dana-Farber Harvard Cancer Center, because between the two, the full space that needs to be spanned to attack cancer is spanned. So the last comment I'd like to make is about the economy meeting that took place last week. It was the awards meeting, a room with 300 plus people were in it, talking about cancer research from different angles, people who made terrific contributions and corporations and so on. One of the awards was for the best big idea in cancer research. There were 5 prospects put up for that best big idea in cancer research. The Bridge Project was the best big idea. With that,
thank you, Art. Indeed, the goal of this unique initiative is to bring together clinical investigators, molecular cancer scientists, and engineers focused on treating the most intractable forms of cancer with the hopes of dramatically improving clinical outcomes and accelerating cures. Quite remarkably, as Art mentioned, it is funded entirely by philanthropy, mostly from individuals and family foundations. We fundraise together with the Dana-Farber. We share the money equally as per Art's vision. And teams have to have at least one member from MIT and one from the Dana-Farber Harvard Cancer Center. And the emphasis from the start of this signature program was funding research that will rapidly translate into the clinic to benefit cancer patients. And so we funded our first teams just over 5 years ago, and we've now had 6 rounds of funding, 37 teams of geniuses from Harvard's cancer centers and MIT's Koch Institute, who together are blending engineering, cancer biology, chemistry, and clinical oncology to create these new solutions for some of the most challenging cancers. In the first 4 rounds, we funded just 15 teams, and then in the past 2 years, thanks to this really generous $20 million challenge grant that we received from the Commonwealth Foundation, we have been able to significantly increase the number of teams we were able to fund. And so we've also added a footbridge category of awards, which are tiny little seed funding of awards, and an expansion bridge category, which is to fund phase 1 clinical trials. And you'll hear from one expansion bridge team tonight. Remarkably, in just over 5 years, which really in science and research, cancer research is pretty rapid, our teams have collectively filed about a dozen inventions, already launched 4 companies, and 6 teams are in or close to clinical application. So tonight we have 2 Bridge Project funded teams who will tell their story. First up is a lung cancer vaccine team. It is made up of 3 presenters. When we sent you the invitation, there was only 2 listed, so we have a bonus. Roberto Chiale received his MD degree from the University of Turin in Italy. He has worked on the ALK oncogene for about 20 years. 5 years ago, he was appointed Associate Professor of Pathology at Harvard Medical School, and his lab is at Children's Hospital. Then Marc Murakami received his MD PhD from Johns Hopkins, did his residency at Mass General, and then his clinical fellowship at Dana-Farber before he started his own lab and in the Low Center for Thoracic Oncology. His research focuses on developing novel therapies for lung cancers with mutations in genes like ALK. And then Daryl Irvin, PhD, is professor of biological engineering, material science and engineering, at MIT. He is one of our newly minted associate directors at the Koch Institute, and his lab is in this building. His research is focused on the application of engineering tools to problems in cellular immunology and the development of new materials for vaccine and drug delivery. Please join me in welcoming our 3 wonderful lung cancer vaccine team members.
Okay, thank you, Anne, for the nice presentation. Thanks everybody to being here and of course for the invitation. So I have the pleasure to start this more scientific part of the night, trying to explain what was our idea a few years ago. The Bridge Project started, as was very well said in the introduction, trying to join the effort of the Dana-Farber Harvard Cancer Center, in this case my lab first, with a Koch Institute lab led by Daryl, which we talk after me. And we first were awarded a Bridge Project, which was a kind of preclinical effort to try to establish the concept of the cancer vaccine against ALK that we are going to discuss. And then we expanded this concept to a potential phase 1 clinical trial, and that's when a third person, Marc, was involved from the thoracic oncology group still at Dana-Farber. So the project deals with the idea of developing a cancer vaccine specific against particular subtypes of lung cancer. And this subtype of lung cancer is basically driven or is because of this particular oncogene that is called ALK. And this is an oncogene that is spread and shared between several different tumors. Here is non-small cell lung cancer, of course. It was originally discovered in this type of tumor that is a lymphoma very frequent in children. And as you can see, the alteration of the same oncogene is spread across different types of tumors, from blood tumors to solid tumors. And the reason why this project started is because all of these tumors have in common one portion. They generate an oncogenic protein, so a protein that drives the tumor formation. And all these tumors have in common one portion of this protein that correspond to a fraction of the ALK gene. And so we thought that this could be, first of all, a very nice common theme among all this tumor. And we decided that we could try to find a specific cure for, in particular for lung adenocarcinoma, because the expression of this oncogene is relatively frequent in lung cancer. Is about 5 to 6% of lung cancer, and actually this is the most frequent gene to be altered by a chromosomal translocation and generate a fusion protein in lung cancer. And there are several new cases, of course, of lung cancer in the US, about 8,000 new cases only in the US, and typically the category of patient affected, maybe also Marc in the clinical part will briefly say about this, is young females, typically non-smoker. And the good thing is that now this tumor can be very well or relatively well treated by a series of specific inhibitors that target the activity of this protein, which is an enzyme, a tyrosine kinase more specifically. And so this inhibitor can block the activity of this enzyme, and by blocking the activity of this enzyme, basically prevent the tumor from growing further. And as you see, for example, here, but we will go back, there are a couple of examples of two inhibitors. One is called crizotinib, which was the very first ALK inhibitor to be developed, and then second generation inhibitor called alectinib, and you see that the survival is much better than before, and Marc will show also that. The problem with this inhibitor is that they are very expensive, and even if they can extend the survival of the patient, really they can never achieve a complete cure, because the tumors, unfortunately, is still there, even if under control by this inhibitor, but practically never really goes away. So there are very, very few patients that are completely cured by this inhibitor treatment only. So we thought that there was need for something better. And of course, we thought about the immune system to help in treating this tumor, and in particular, in this circle that explain how the immune system recognizes tumor cells and generate some response that can help to contain the tumor growth. We are focusing on this part, which is basically the part in which the tumor antigens, so the protein that are generated by the tumor, are presented to the immune system, and they prime the cells of the immune system, in particular T cells, to recognize antigen specific of the tumor. So this portion is typically involved in the priming, and is typically a concept of the cancer vaccine associated with this part. And the idea stemmed for a series of considerations by which this particular oncogene is very amenable for this type of cancer vaccine approach. First of all, this is an oncogene that is expressed only by tumor cells and is not expressed by normal tissues. This is good because in principle, the immune system can recognize this protein as novel, generated by the tumor, and not present in normal cells. Here, for example, you see an ALK-positive lung cancer. On the side, you see in brown are all the cells of the tumor that express this particular protein. And on the left, for example, is an ALK-negative tumor, and surrounding, you see normal cells that clearly do not express the protein. Another key point is that this oncogene is expressed in all the cells of the tumor. So ideally, if we were able to develop an immune a cancer vaccine that could target and really recognize this protein, in principle, we could attack and destroy all the tumor cells, because all of them, they really express the antigen. And then, of course, another key point is that this oncogenic protein is essential for the growth of the cells. For example, here you see a lung cancer tumor before the treatment with ALK inhibitor, and after treatment you see that the tumor disappears. It means that these cells are really dependent on this tumor, so there is a very little chance that tumor cells could escape without expressing this protein. And that's very important because we want a cancer vaccine that can really target all the cells and prevent the escape of negative cells. And a final key point that really kind of completed the essential part that we needed to move forward in this idea is that we needed also to show somehow before generating a cancer vaccine that this protein is indeed recognized by the immune system as a novel protein produced by the tumor cells. And the evidence came in lung cancer patients by analyzing immune responses specifically against this protein, and we were able to show in collaboration with Marc that only ALK cancer patients have a high titer of immune responses against this protein, particularly either antibodies or T-cell responses. So this is also an essential requirement, because by just analyzing patient samples, sera or cells, we were able to show that already the immune system in a sort recognized ALK as a foreign antigen. So we decided to develop this concept, and we tested first in preclinical model. And here is just one single slide to show these are mice, so they are not human, engineered to develop lung cancer that are driven by the same oncogene exactly as in humans. And you see, for example, here is a typical development of lung cancer in a mouse driven by this oncogene. So it starts with one tumor, and then there are many tumors until the entire lung basically is filled up of different tumors. And when we vaccinate these mice with an ALK vaccine only, so only the vaccine without any other therapy, we were able definitely to contain, not completely eradicate, but definitely to contain the development of the tumor. And there are some data. So based on the concept idea and based on the preclinical data, we decided to start this project, and we joined with Daryl, because he had a wonderful technology to develop probably the best vaccine approach possible. And I'm handling to Daryl.
Okay. So now where I come in is sort of rewinding to earlier in the story when Roberto was discovering the potential of targeting ALK with a cancer vaccine. And this data we're showing here is in a mouse model where you can just inject DNA encoding the target antigen and get an immune response that allows you to say that, yes, a vaccine against this target could be good. Unfortunately, injecting DNA into people doesn't elicit nearly the same strength of immune response. So to go into the clinic, we knew we would need a different kind of vaccine. And that's where my group came in. We'd been looking for ways to make very potent cancer vaccines that could be used in humans. And I had a postdoc come to the lab about 5 years ago now, who got interested on a type of vaccine called a peptide vaccine, which is very simple. You take a portion of the mutant protein that you want the immune response against, just a peptide, and mix that with an inflammatory agent and inject it. And this is basically the same idea of all the vaccines you get as kids, except now you're injecting, instead of a virus fragment, you're injecting a piece of this mutant protein. And this was known that this could elicit T-cell responses in humans, but they're generally very weak. So the problem that we had to solve is, how do you make this kind of vaccine work much better than it does? And for a peptide vaccine to work, when you inject this into the muscle or into the skin, all of the action needs to happen in the downstream draining lymph nodes. So the vaccine peptide needs to be carried through lymphatic vessels to the lymph nodes, wherever they are locally, to that injection site. And Haipeng got very interested in the question of where vaccines go when you inject them, because he would always say, I'm just a chemist. And he would say, so as a chemist, he would say, what's the physical chemistry of where these molecules go? And we were shocked to find that in mouse models, if we injected labeled vaccines, took the lymph nodes out and looked by fluorescence imaging to ask where the vaccine went, that almost none of it was actually getting to the lymph nodes. And this is the positive control, where we inject the whole vaccine dose into the lymph node outside of the animal. So this told us that we've got one problem that we can clearly try and solve, which is get the vaccine to the place it needs to go. Now, we also drew inspiration knowing that there are some clinical procedures that deliver things very efficiently to lymph nodes. And one example is sentinel lymph node mapping, where in breast cancer or melanoma patients, when the surgeon resects the tumor, they may inject into the resection site a dye like this Evans blue dye that has the property that it goes very efficiently into lymphatics and goes to lymph nodes and then stains them optically so the surgeon can find the lymph node in the fatty tissue and resect it to look for metastases. And these dyes were originally discovered empirically, but they've been around long enough that it's now understood if you want to make a great sentinel lymph node mapping dye, you make a compound that binds very tightly to albumin. So we have these sort of two issues. Peptide vaccines are not going to lymph nodes. These sentinel mapping dyes go to lymph nodes very well, and why is that? And it basically has to do with the physiology of solute transport in tissues. So when we inject a vaccine or any other drug into the tissue, that drug can either go into the blood vessels or it can go into the lymph. And we want our vaccine to go into the lymph. For small compounds, low molecular weight compounds, they can pass through the basement membrane and the tight junctions between endothelial cells, and they're going to go into the blood because the blood is flowing tenfold faster than the lymph. But larger macromolecules and particles tend instead to go toward the lymph. And this sort of explains the peptide vaccine is not going to the lymph because it's too small, basically. It all goes straight into the blood and gets lost in your circulation. Whereas if you look at accumulation of protein in the lymph versus molecular weight or size, then the larger the macromolecule, the more likely it is to go to the lymph. And albumin, anything that binds to albumin is up here in the plateau going one way from blood to lymph. Albumin is a large enough protein that it all goes to the lymph. And so that inspired the vaccine concept. What if we make a vaccine that copies what these sentinel mapping dyes do, make a vaccine that binds to albumin, and thereby get chaperoned to the lymph node, sort of hitchhiking on albumin. Because one of albumin's main jobs in vivo is to serve as a fatty acid transporter, it has 7 different binding pockets for lipids, we thought that we could make such a vaccine by taking our peptide vaccine and conjugating it with a phospholipid tail that would bind to albumin on injection and then deliver it down to the lymph node. And it turned out that this idea works tremendously well. So in mice, if we inject labeled vaccine and then take the lymph nodes out a day later, if we vary the length of this lipid tail, the longer the lipid tail, the more tightly it binds to albumin, and the more vaccine we see arriving at the lymph node. And by dialing that in, we could show that you could take a vaccine that had no ability to accumulate in lymph nodes, and here the vaccine is labeled green. You see it's not getting there, but if we put this albumin binding tail, now it's impressively accumulating in the lymph node. And we've recently, in part supported by the Bridge Project, showed that this also works in large animals, because a big concern moving to humans was mice are very small. Is this just an artifact of immunizing these small animals? But here we see the same result. If we inject a traditional peptide vaccine, so this is the labeled adjuvant and this is the labeled antigen, the peptide, in the lymph nodes of rhesus macaques, and you see the vaccine's not getting there, but when we put the albumin binding tail, both the antigen and the adjuvant go nicely to the lymph nodes. And it was around this time that we had shown that this idea works that I met Roberto. I was giving a seminar over at the Dana-Farber for the immunology program, and he explained to me the ALK vaccine idea. So we realized immediately that this was a great partnership to take his validated target and our vaccine technology and put them together. But of course, nothing can happen in research without funding, and the Bridge Project was a really important catalyst to allow us to actually do the experiments. And so the experiments were to first make an ALK form of this vaccine and show that when we immunized and looked for responses to different parts of the ALK protein, that we could see helper and killer T-cell responses elicited when we vaccinated mice. Shown here, we're showing that the T-cells vaccinated against this antigen are making important cytokines. And then going back to the model that he showed earlier of a mouse model of ALK-positive lung cancer, and showing that a peptide vaccine could do about as well as the DNA vaccine did earlier, except now we believe this peptide vaccine can also work in humans. And which brings us to the next stage of what Marc has to say.
Thank you very much. It's a pleasure to be here. And I think, even though I did my residency training at Mass General, my fellowship at Dana-Farber, even though I live a few blocks away in Central Square. I'd actually never been to the Koch Institute before until it was the Bridge project, expansion bridge grant funding that came along. That's what led me to meet actually Daryl and work with Roberto. And so I think already part of the efforts are very successful at joining the different institutional efforts together. So I'm very appreciative of these opportunities. So, I think we're very excited to translate some of the laboratory findings from Daryl's lab and Roberto's lab and try to develop this into a new therapeutic treatment option for patients with ALK-positive lung cancer. And as Roberto mentioned, there are some very good and effective therapies for ALK-positive lung cancer. There are several different oral tyrosine kinase inhibitor pills that are ALK inhibitors that can very successfully and in a large number of patients shrink down the cancers. One of them being crizotinib, for example. Here again is another example of a patient with lung cancer driven by this ALK protein. After starting crizotinib, the cancer can shrink down very dramatically, and it works in the majority of patients with this type of lung cancer. But the challenge is really the development of resistance. So for patients who start crizotinib, it's usually an average of about 8 to 10 months for most patients before we start to see the cancer grow again and progress and spread to new sites of the body. Some of our more potent ALK inhibitors, like alectinib, are more potent, more specific, high rates of response. But one of the challenges is still that these cancers can develop resistance to that drug as well. It's kind of analogous to the early days of HIV or tuberculosis treatment. If you treat these infections with just one drug at a time, it can work transiently, but then a virus or like cancer can develop resistance to these single therapies, and we really do need to come up with more novel ways of attacking these cancers. So you can see here the curves on the left are what we call progression-free survival. So you can see that compared to chemotherapy, crizotinib slows down the time or the rate of cancer progression, but over time, cancers will develop resistance. And so you can see that the time to progression is about 7 months, even with our more potent ALK inhibitors. We know that there's a lot of excitement about immunotherapy for lung cancer, the class of medicines called immune checkpoint inhibitors, PD-1 pathway inhibitors. These are drugs like Keytruda, which is pembrolizumab, or Opdivo, which is nivolumab, and others. But in ALK-positive cancers, which tend to occur in never smokers or light smokers, these therapies tend to work in a very small minority of patients, probably 10% of patients or less. And we're not sure why the immune system is not capable of recognizing these ALK-positive lung cancers. But our hope is that through vaccination, we can really increase the likelihood that a cancer will realize that the ALK mutant protein in the lung cancer doesn't belong there and really engage the immune system in fighting against that cancer. So we've crafted a phase 1 clinical trial to get the vaccine into patients. And one strategy is just to take different portions of the ALK protein and chop it up into smaller segments and vaccinate a patient with all the different fragments of ALK. That might be 20 to 30 different pieces of that ALK protein. We're also trying to look to see, are there really only specific fragments of the ALK protein that are most likely to stimulate the immune response? And this is one technique that we're using. We're in collaboration with Kathy Wu's lab at Dana-Farber. We're engineering cells to express different portions of a human's antigen presentation machinery, the HLA or MHC molecules, in conjunction with ALK fragments to say which are the fragments of ALK that are most likely really to stimulate that immune response, and can we tailor or narrow down our vaccine, or even more personally tailor the vaccine to each individual who might have a different immune system or a different HLA background. So this is some of the work that we're doing, looking at the immune peptides, the specific sequences that are most likely to be presented to really pick out the winners for our vaccine development. And this is a scheme of the clinical trial that's being developed and funded in large part through the Expansion Bridge Grant, where initially we will test the amphiphilic conjugated peptides, the technology that Daryl's lab has developed, alone, just to make sure that in patients, since this is the first time this new therapeutic will be administered to humans, we want to make sure that it's safe, and we think that it will be. And then after a few patients are enrolled, after we've established the safety, then we'll move into administering it to patients either in combination with some of the immune checkpoint inhibitors or in combination with some of our oral ALK kinase inhibitors. And we have different vaccine schedules for vaccination, priming, and boosting phases here, along with blood draws and immune analysis of these patients to really make sure that this concept is producing the effects that we want in our patients. So we want to be able to draw blood, analyze the immune cells both in the blood and from the tumor to really understand, are we generating this immune response? Are we getting the immune cells into the tumor, and are they actually fighting the tumor? So we're very excited about this opportunity, and we will be hoping to open this trial very soon at Dana-Farber. So I know that all of us here are also very appreciative of the numerous people, both in the lab and on the clinical side of things, that have been working to develop this and make this a laboratory-based concept and translate it into a clinical trial reality. So with that, we can stop and take any questions.
Thank you so much. So we have time for a very brief couple of questions because we have a whole other story to come. Who wants to ask a question? Please.
Hello. Hey, I had a question. Since this ALK gene is present in many other tumors too, do you envision there to be a difficult transition from the lung, or is this vaccine going to be readily available for other type of tumors? Fairly readily.
You should all have your mics working on your...
Thank you.
Is it on? Yeah.
Is this okay? Everyone's here. So I think that's a great idea. As Roberto mentioned, these ALK rearrangements or even ALK mutations can occur in a variety of tumor types. It's been described in lymphomas, in colon cancer, in other type of sarcoma type tumors. So I think the potential here is much broader and can be applied to any number of tumor types that are dependent on this protein. So I think we're quite excited about developing this first on a smaller scale and then rapidly expanding if we see a signal to incorporate this into other types of cancer.
Hi, over here. Thank you so much. It was a great talk. Curious, I'm sure you're all aware that one of the major challenges in lung cancer is brain metastases. And so I'm wondering if you can comment, at least in your preclinical models, whether or not you understand the ability of the vaccine to actually benefit sort of a brain cancer, metastases or a model like that.
Yeah. So in our model, we could not test, in particular, brain metastases because our model does not generate brain metastases. But however, there are other examples by which similar cancer vaccine really can be effective in the brain. So the reason why is possibly because during the metastasis, the barrier that separates the brain from the other tissue is kind of changed and altered, so there is more possibility for cells of the immune system to enter the brain and be active there. Yeah.
I think we also, I think it is a particular challenge, especially in ALK-positive lung cancers and in other lung cancers. We know that with some of our immunotherapies, these immune checkpoint inhibitors, we're not sure exactly why, but we often see that if a patient responds systemically to these immunotherapies, they often see an intracranial response. So we might also see brain metastases shrink, and we're not clear if the actual antibodies that therapies are getting into the brain or if it's generating an immune response in the body and that those immune cells are actually getting to the brain. So we're hoping that if we can stimulate the immune system in some way to recognize the ALK-rearranged lung cancers, that we'll also hopefully see a response in the brain as well.
Oh, Linnea has a question.
Will your phase 1 trial only be at Dana-Farber, or will there be other sites as well?
I think we're initially opening it within the Harvard Cancer Center, so we actually just had a meeting with Alice Shaw at Mass General yesterday, and I know she's an enthusiastic collaborator. So I think initially it will probably be open at this individual site just because I think here we have the kind of scientific tools and expertise to be able to analyze the immune responses, and then our hope, if we see some promise, would be to try to expand it to other sites.
Okay, final question at the back, I think, and then afterwards you'll just have to grab these three in the galleries. Oh.
Thank you for the very intriguing talk, and look forward to the results. Some of the checkpoint inhibitors have had a side effect of cytokine storms. Do you see that as a potential safety issue with your approach, or is that something we don't have to anticipate?
So that's a very important question. So these immunotherapies, there's a lot of promise, but also a lot of toxicities, these immune-related adverse events. So, you overstimulate the immune system and you can get inflammation in really almost any part of the body, some of which can be, very serious. I think our understanding of vaccines that have been used in other clinical settings is that generally they're well tolerated, and you don't see a lot of general kind of overstimulation of the immune system. I think we're trying to tailor this vaccine so that we are really only stimulating anti-ALK responses and not stimulating responses against other potential proteins that might cause side effects for patients.
All right, well, thank you very much. All right, so next up, a team led by David Weinstock and Scott Manalis. Now, David Weinstock is Assistant Professor of Medicine at Harvard Medical School, a staff physician and independent investigator at Dana-Farber Cancer Institute, where he directs a translational research program focused on novel therapeutic targets in lymphoid malignancies. Scott Manalis has undergrad degrees and in physics, PhD degree in applied physics, and joined MIT as a faculty member in 1991 in biological engineering and mechanical engineering. In 2011, when the Koch Institute opened, he was one of the 26 faculty members who moved his lab in. He uses conventional silicon processing techniques to fabricate fluidic devices and exploit the unique physical properties associated with micro and nanoscale dimensions for developing precision measurement methods. He basically weighs things at a really small scale.
We're going to try a slightly different conversational format where Scott and I are going to go back and forth. And please do feel free to interrupt if anything we say is either too sophisticated or too pedantic or not clear. Oh, no, I'm supposed to go first. Okay.
We scripted it.
Yeah, no, that's right. Remember? I forgot.
We practiced hours.
All right, yes. All right. I'm Scott Manalis. No, I'm David Weinstock
right.
from the Dana-Farber Cancer Institute. I'm a leukemia lymphoma biologist and a clinician. And this is Scott Manalis. And you want to go to the next slide?
Yep.
And so we really want to drive home the point that technology transforms cancer research. And there are many different places that can rush to be the first to discover something, to develop a drug against a target, and so on. But transformative advances really come when something like PCR in the 1990s, or next generation sequencing in the 2000s, or CRISPR and gene modification in the 2010s allow you to do something completely different. And that's really what the bridge is allowed. So next slide. And so I take care of patients with acute myelogenous leukemia, AML. This is clearly a significant problem, and so this is United States SEER or epidemiologic data, and you can see that we're not making any headway. So the number of cases, if anything, is increasing with our aging population, and the number of deaths per year is staying exactly the same or increasing. And so the percentage of patients who are dying of this disease is staying the same. It's in the 80% range. Most of those patients are dead within a year after diagnosis. And this is just totally unacceptable. And there have been many, many, many new drugs developed and really no significant advances. So we need something transformative and novel. Next slide. So we know, despite this fact, that AML is actually extremely responsive to chemotherapy. So if you give a patient multiple cycles of chemotherapy like Marc does for lung cancer, and they have a 50% reduction in the size of the tumor, that's considered a success rate. But we know that 80% of patients who are treated for AML will actually have a complete remission. And that's a 99.9% response. So 99.9% 3-log reduction in the total amount of tumor. The problem is the overwhelming majority of those people who go from 1 trillion leukemia cells to 1 billion leukemia cells, we don't cure the additional billion, and they relapse. And once you have relapsed AML, that's almost a death sentence. So there are some patients who are salvaged by allogeneic stem cell transplant, but as you've seen overall on a population basis, we've really made no headway. And so the question is, could we start targeting that minimal residual disease, that very small amount of disease that when you look in the bone marrow or the peripheral blood of a patient, you actually can't see it, but if you use molecular tests and strategies for isolating very rare cells, you could start to directly target those individual cells. So why is that important? Well, partly it's because there are just simply fewer cells to cure, right? But also because we've learned over the past few years, and this is data from Washington University in St. Louis, but we've generated similar data out of my group and others, that cancer is a heterogeneous disease, and I think you all probably know that. So it's not exactly the same cell copied 1 trillion times. There are different mutations within different clones. We call these subclones. And you've got to cure all the subclones. And so when you treat a patient, they've actually got multiple subclones. They go into a remission, and you markedly reduce the number of cells, but multiple subclones survive and then relapse. And so the real question is, what's going on in that minimal residual disease? What are those subclones, and how do we directly target them? So that's the obvious question, right? No one in my field would say that that's not the most important question in the field. The problem is we simply didn't have technology to be able to do that, because if you sequence these with next generation sequencing, you can find all kinds of mutations, but we have almost no drugs that we know are directly linked to those mutations. So in some contexts, IDH inhibitors are an example, or FLT3 inhibitors are an example. If you have an IDH mutation, you can give an IDH inhibitor, you have a much higher response rate. Same thing as an ALK rearrangement and an ALK inhibitor. Same thing with a FLT3 inhibitor. But all those other drugs, and we have lots of new drugs coming down the pike, we have no idea who to give them to. It's completely empiric, which means we just give them the next one on the list. And so we need a smarter strategy to be able to say, this is the right drug for this person at this time, precision medicine. And specifically, could we do this in an iterative way? So could we get a patient into a complete remission? Now they've got this small amount of disease, and I won't do the green light thing that makes me nauseous, and pick the right drug and then reduce their amount of disease, and then do it again and then pick the right drug and reduce their amount of disease, and so on. And maybe even, I have a question mark there, because maybe even that could lead to cure. But we've got to have a test that can actually allow us to analyze single cells that are extremely rare and understand what would be the right drug for that. And so that's where Scott comes in.
Okay, so that question of finding the right drug, what we'd like to do next is show you that diving board, like what you see on the screen, could be a reasonable approach to pursue for looking at the next best drug. And what this diving board does is it weighs individual cells with an incredibly high degree of precision. About 1 part in 10,000th the weight of the cell is roughly the noise of the devices. So very small changes of weight of a cell, it can resolve. And the way it does it is it's vibrating, kind of in the same way a diving board on a pool vibrates. And if you go out to the end of that diving board and you shake your knees, you'll be vibrating up and down. As soon as you jump off, what happens to that board? It starts to speed up and vibrate at a faster speed. And we've made this one hollow, and so it's vibrating as a cell flows through it. And when it comes to the very end, and I will use, hopefully this doesn't make David nauseous. David, when you look at the very end like this, all right, you see a dip in that, and that is like jumping off the diving board. Okay. And so that is the weight of the cell, and you can imagine then weighing that same cell again and again and again. And the cell, if it's in media with nutrients, it'll grow and it'll continue to accumulate mass. And you could actually look at how its weight changes over time. And so around the time that met David, about 5 years ago or so, what we were doing, Mark Stevens in my lab, a PhD student in the biology department, was using this tool in a collaboration with Matt Vanderheiden here in the Koch Institute, who studies cancer and metabolism. And here's actually what the weight of these cells looks like. So each little point here is like one of the dips going through that device. And so you can see this cell is accumulating mass, kind of here's 49 picograms, 50, et cetera. And the question we were looking at with Matt is, what does the cell eat? Turns out something that has never really been measured in a direct way. And so what we thought is that we can have cells in media and then deplete something like glucose and see how that changes its trajectory. And you can see when you actually reduce glucose here, the slope of this line actually changes. It actually goes down. So because it can't consume glucose like it could before. So this is a long project. We won't talk about it all this evening, but this just led then to the question, what about a cancer drug? Instead of depleting glucose, what if instead you expose that cell to a cancer drug? What could happen on this very short time scale that we're measuring? Maybe the drug is exposed to it for a few hours, but it's a very sensitive measurement to look at how it could change the cells. And so that's what we began to look at, and the bridge really brought us together around this question, the problem that David posed to you. We just found incredibly exciting, and my students and I really wanted to work on it. And so we started to do that then. And so Marc paired up with Marc Murakami in David's lab to work together on this idea. And so what they did is they took a cell line that has a BCR-ABL translocation into it, so it's sensitive to imatinib. And then they treated that cell line for different times. And then at these different time points, just ran these cells through the device and weighed them and measured the rate of change of their weight, like that little slope that you saw. And so what's plotted here is that slope of the line. We call that this mass accumulation rate. It's just what that slope was on this previous slide. So we're plotting just this slope here. And so here are the cells untreated. And if you treat them after about 8 hours, you can see that they're not accumulating weight anymore. And they're also pretty small. And kind of in between, in just a couple of hours, there's a change in their slopes. So this was encouraging to us. We really thought that then we could now try more interesting types of cells and more interesting types of drugs. But we had a big problem. If you look at the number of dots on this screen, each dot is a cell, there's a couple dozen, and that took like 3 months. And the reason why is that each cell had to be kind of weighed independently. And so it's a very laborious process of loading in the cell, trying to capture one, having the pressures kind of adjusted. It's kind of like juggling, I'm told. I've never done it in the lab, but from what my students say, you have the cell go back and forth through this thing to get weighed. And so it's laborious. And so this, we needed a solution to this. We needed this to be high throughput and easier to use, and then we wanted to test this. This is where the bridge came in. So we thought, with the bridge, can we solve this problem? Can we make it easier and higher throughput, and then really channel it towards more interesting experiments? And so this is how we solve the problem of throughput, using arrays, which is obvious. But what's less obvious here is that these devices are connected in a serial way. There's just one very long channel with these devices strung along the way. And so a cell gets weighed, and then it spends some time going through these delay channels until it gets weighed again. And it takes about 20 minutes for a cell to go through this. And during that time, it gets weighed a dozen times. And then we can get that slope all at once. But now we could have many cells go through this at the same time. And we don't have to do this juggling act. We could just pressurize it, drive the cells through. And if you watch this a little longer, you'll get really tired and start to sleep.
Yes. Actually, it's very interesting to watch the audience because I can see your eyes going up and down.
When I have a tough night, I go right to this slide. It's very, very, very powerful. So this is what the response. Okay, so this is actually what the device looks like. It's made with kind of all the techniques that are used to make computer chips. It's called microfabrication or MEMS. This is made out of silicon, kind of patterning with light layer by layer. And this is these delay channels. If you zoom in here, you can see these are the little diving boards that are all connected. You notice that there are different lengths. This is a shorter one. This is a longer one that makes them vibrate at different frequencies, and that's how we read them out. And we make them now in France with a really talented team there that can make things at scale. And so they're made on 8-inch wafers. We make hundreds of them on a wafer, and a run will do thousands. So this is completely manufacturable now. We can just order and design these things. And this is what the instrument looks like now. This is the latest version that folks in my lab, particularly Salim Okum, has made. that now is kind of benchtop size. All the electronics, the computer, and everything is in this box. This is this little chip. If you zoom in, you can see the chip there. And so that's what the system looks like. So this is what the results look like now. So if you flow through these cells, this is what comes out. And so each one of the little trajectories here are the growth of individual cells. The blue color here is the first one, and the red is towards the end. And so what's on this slide here happened in the course of, well, I guess 200 minutes, but that was 3 months now that Marc and others in the lab could see in 200 minutes. It was very, very satisfying. And so then, now what happens when you apply drugs at this sort of throughput? And David is going to show you some of the things that we measure.
Sure.
Oh, let me just make one more comment.
Go ahead.
This is off script. All right. So Marc at that time, there's a separate problem. Right about the bridge was starting, Marc was ready to graduate. This was like the last thing he did for his thesis. And so he had all the expertise to kind of run this machine and do these measurements, pretty specialized. So David and I were able to convince Marc to stay. Not as a postdoc, it wasn't really a postdoc position, but it was kind of a unique position where Marc became fluent in both of our labs and allowed our team members to kind of work together in a way that turned out to be absolutely crucial for being able to make our project work. Okay, so.
No, I think so, and trying to find funding for a person like that who can literally spend half their time in one arena versus another, it's just extraordinarily difficult. I would also take a step back and say there is a long and checkered history around the idea of taking patients' cancer cells out of their body and then exposing them to drugs and then trying to tell a patient you should get drug A or drug B and so on. And that has largely been a failure for two reasons. One is that the drugs that we had in the past just didn't work that well. Right? So if you added cisplatin to a lung cancer, you might be able to show that that cancer cell died, but we knew that the response rate was 25% or something like that. Now we've got drugs that can reduce the amount of disease 99.9 or even more logs than that. So we've got these very active drugs with Boolean responses. It either does work or it doesn't work. And the second is that, as Scott showed, we can now read things out in a matter of hours, and sometimes even shorter than that. And so we're overcoming many of the factors that really plagued some of the previous in vitro systems. We had to grow cells for days, and things just were no longer predictive of what would happen in a human being. So this was tremendously exciting to me. This is data showing, again, in these cell lines that have BCR-able rearrangements, and so they either should or shouldn't be sensitive to imatinib, that the data that Scott was previously able to generate in his lab over the course of months, we could generate in hours, and we actually set up a CLIA space in the Brigham and Women's Hospital. So this is a space that has special capabilities and satisfy certain requirements, so you could actually report the results of an assay directly within a patient's chart. We set up these SMR devices along with next generation sequencing and other approaches, all in tandem, so we could try to understand what the results would be. And I'm not really sure what I'm supposed to say here.
You made that point earlier.
Okay, good.
Skip that.
All right, great. So, and so this is now the way the data looks, and you can see with very short incubations, 2 to 4 hours, 4 to 8 hours, and so on, we can take these cells and we can understand should they be sensitive. In this context, on the left, where you have BAF3 BCR-able, they should be sensitive to imatinib, and so you can see as a population, the mass accumulation rate goes down as a function of mass. And on the right, where you have a resistance mutation, T315I, that should prevent sensitivity to imatinib, you don't see activity of imatinib, but you do see activity from another drug called ponatinib that we know is active against these cells that have the T315I mutation. So we've gamed the system a little bit here, but we've been able to validate that it acts as predicted. And so one of the really important next steps for our work was to move into patient-derived xenografts. So my lab has really led an effort at the Dana-Farber and Boston Children's and Mass General to try to develop models where we take human leukemias or lymphomas or other tumors and we grow them within very immunodeficient mice. And so now we can test drugs in those animals and understand how will a human leukemia or lymphoma respond to those drugs. And in the context of these experiments, we can make predictions with the SMR. We can take the cells, we can put them in a panel of different drugs, we can say this would be the right drug, this drug would not work, and we can actually test whether or not the SMR is capable of predicting those actual responses. And this is absolutely essential for being able to then advance into human beings, because we wouldn't want to take a test like this into human beings where we're telling a patient, you should get drug A versus drug B, if we hadn't already proven it on human leukemias. And so this is just an example of an experiment that we did in these mice. And this is using, as you can see, 25 microliters of blood. So I know you're all MIT alumni or so on, so you know what that means. But I like to say that it's 1/200th of a teaspoon of blood. So that's the amount of blood that we're dealing with. And we can then assay very small numbers of cells, in this case 21 in the vehicle versus 31 in the drug, and we can see that these cells in fact are sensitive to that drug. And so this really is the proof of principle that we can move into settings where there's really minimal amounts of disease, isolate very, very rare cell populations, and then understand what they might be sensitive to. And this is an example of its application in a human being. And so this is a patient who was taken care of by my colleague Dan D'Angelo, who has AML and unfortunately has relapsed multiple times after different therapies, and went on to a clinical trial with a Novartis agent called HDM-201. And so we isolated blood both before the patient started drug and then 48 hours into treatment. And here we didn't expose the cells to any kind of drugs before running through the SMR. We just said, what do the cells look like before the patient gets the drug? And what do the cells look like with the drug on board in the patient for 48 hours? And so you can see the black cells are the cells that we isolated prior to the patient getting the drug. And you can see essentially all of them are growing. They're all above that 0 point. And that tells us that even when we isolate these cells and we bring them out of the body, they're still growing. And we should be able to see then a delta if we apply an active drug. So 48 hours into therapy, that's very, very early. The patient had manifested no response whatsoever. Their white blood cell count was the same. Their blast count in the peripheral blood was the same. We had no idea whether the patient was going to respond. But you could see already there were, in red, a significant fraction of cells that were starting to die. And this is an assay that could pick that up without really anything else determining that. And I shouldn't say they were starting to die. They were losing mass. And our prediction is that those cells would lose mass. And so the most important question then from this kind of data is what's the difference between those red cells at the bottom and those red cells at the top? What's the difference between the cells that are now losing mass and presumably are going to die and those cells that aren't losing mass? And can we understand by a much more careful analysis of those cells what are the subpopulations of cells that this drug might not be working for, and how do we create novel combinations and so on?
Yeah, so that question we wrote that question up in terms of a center grant, which after many tries got funded. But
not 12.5 million tries.
And really what this is about is sequencing these cells, the transcripts of the cells, to figure out their transcriptional, the gene expression of these cells, to better understand, as David said, what makes these cells different. So this has just started this past summer. It recruited now a bigger team at the Farber and at MIT, and very excited about pursuing this. The other aspect is really being able to bring this towards the clinic to ask what is the next best drug to treat the patient with. And for this, David and I, together with others, have started a company called Trevera. And this company, the principal people in this company, Mark Stevens, who you saw previously, Salim Okam, who is kind of the architect behind this whole system, and Rob Kimmerling, who's also made very important contributions. These are the three guys that were largely trained through the Bridge program that will go off and start the company. We have a CEO that has a lot of experience in genomics that got very excited about functional assays during that time. So we're very excited about that. And then finally, we just want to deeply acknowledge the Bridge. It just was a tremendous opportunity for us. So perfect. Thank you.
Which is which?
You decide. Everyone is just either desperate for dessert or...
So how much does the weight capacity, increase in weight capacity, correlate with the division capacity?
Yeah,
so is there a direct correlation or just cells that are growing?
If it's a cycling cell, like a cell line, it's directly related. So we could actually, in 10 minutes of weighing that cell, we could know how long it will take to double. But for primary samples, sometimes they may not be in the cell cycle. And when you take them ex vivo, if they were in the cell cycle, they might behave differently. And so when we do our ex vivo measurements from patient samples, we're really interested in the differential between what they were doing before and after the drug.
Anybody else? Art?
So this is more of a comment than a question, but I do want to say I'm thrilled by this project. When I originally thought of bringing these communities together, this was precisely the kind of thing I envisioned in a very qualitative way, bringing a new technology to the table and having it potentially change the course of the attack on the disease. So I am absolutely thrilled with what you're doing, attograms and all.
Thank you, thank you.
Thank you. And we should thank you as well, Art. I think there are people who have truly innovative ideas, and there are people whose checks clear. But there's very few people who do both.
A last question, because we're a little behind schedule.
So you've shown the application here for blood tumors. Do you have a vision on how you would extend this to other tumor types, and including samples from solid tumor biopsy?
Yeah, so it's a good question. Obviously, the solid tumors are a lot harder because you have to think in one way you can go and disassociate them, and people have done that to do like transcriptomic measurements. But there's a lot of risks associated with that because you could change the response of the cells. The other way, though, is to consider circulating tumor cells. And so one of the things we're really pushing in the lab is to be able to take, now David showed you thousands of cells, we now have that down to hundreds. And I think in about a week, in about a year or so, we'll have it down to tens of cells. And so all these fancy ways that have come out to collect circulating tumor cells, and now a lot of these labs are growing cell lines from these, but with those fancy collection methods, which this is not high throughput to enrich from blood, but if you take those other ones from other labs and then enrich and have these 10 cells, we could think about drug testing them. And so that could be an entryway into the solid tumor.
But you've also got clumps of cells and organoids.
Yeah, that's a good point. Yeah, so that's the other. So maybe you don't digest all the way into single cells, but you go just part way into kind of clumps. And then there's also patient-derived organoids. So there's other ways that you could get at that and still keep the microenvironment and be able to drug test that whole entity, like just a surrogate for the tumor. So I think there's a few different strategies to that.
All right, thank you so much. I hope...
Thank you.
I hope you enjoyed this evening as much as I did with the remarkable stories of success and accelerated progress that can happen when you can truly bring two powerhouses together to fight one of the world's biggest health challenges. So please join me in a round of applause for all of our speakers tonight, as well as I also want to take this opportunity to thank to express my admiration for our Harvard and MIT trainees, for our staff, for our fundraisers, and our administrators who have really worked together across this sort of institutional boundaries to make the Bridge project a huge success. And of course, last but definitely not least, I want to express immense gratitude to all of our Bridge project donors, and of course, especially Art and Linda Gelb, who really kickstarted this incredible initiative, this incredible partnership, and the story of the Bridge project, a story of hope and inspiration of biology and technology leading towards rapid clinical translation for urgent clinical needs. Our next Solutions with Insight will feature our other signature program, the Frontier Cancer Research program, and to round off this year's theme of highlighting our signature programs here at the Koch Institute. At Cells in the City, you will hear the story of two remarkable Koch Institute faculty startups, Squeeze and Lumicell. Thank you for coming. I hope to see you again soon. Please find some desserts in the gallery as well as coffee. Thank you for coming.